academicians, researchers, scholars, and participants joining us from different parts of India and abroad. It's my immense pleasure and privilege to welcome you all to the SPR Fellow and Best Doctoral Thesis Award Lecture Series, organized by the Society for Plant Research, Uttar Pradesh, India, on 29th and 30th through online webinar mode. This prestigious lecture series has been conceptualized to recognize and celebrate excellence in the field of plant science, biotechnology, agriculture, environmental science, and allied disciplines. The event brings together outstanding researchers and academicians whose scientific contributions have significantly enriched the scientific community and society. At the outset, I extend my sincere gratitude and regards to Professor Dr. S.K. Bhatnagar, sir, founder, chairman of SPR, and my supervisor too. Professor R.R. Chilani, sir, immediate past president and chief patron, SPR, and Professor Banna Bose, ma'am, president, SPR. Their visionary leadership and continuous encouragement have made this scientific platform highly impactful and inspiring for researchers across the country. I would also like to acknowledge the dedicated efforts of the organizing committee, screening committee, technical team, and especially Sri J. Narayan, coordinator, SPR Awards and technical head, for the successful organization of this webinar. The SPR Fellow Award honors distinguished scientists and academicians for their exceptional contribution in research, innovation, teaching, and scientific advancement. Likewise, the Best Doctoral Thesis Award recognizes young researchers for their outstanding doctoral research and academic excellence. Over the course of these 2 days, we shall have the opportunity to listen to enlightening lectures and scientific presentations delivered by eminent awardees representing premier institutions such as ICRISAT, ICR Institute, Central Agriculture Universities, Aligarh Muslim University, Utkal University, University of Delhi, CSIR institutes, and several other renowned universities and research organizations from India and abroad. As a moderator, it will be my privilege to facilitate today's session and coordinate the smooth flow of the program. After these brief introductory remarks, I shall invite the awardees one after another according to the schedule sequence provided in the program. I request all the participants to kindly keep their microphones muted during the lectures and use chat box for queries and interactions. Question answer discussion may be taken up after each lecture or at the end of the session, depending upon the schedule and availability of the time. Now, with great respect, I invite Professor Dr. S.K. Bhatnagar, sir, for the introductory session. Sir, over to you.
Thank you, Dr. Jyoti, for briefly summarizing the start of this very important lecture series. At the outset, let me congratulate all the fellow awardees for 2026 and Best PhD Thesis awardees for 2026. As you all know that during 2026, the executive body decided that we should recognize the younger students who have submitted their PhD thesis in the recent years, and by introducing this very important award, we were in a process to acknowledge the contribution of young fellows who are basically the backbone of science in the time to come. Fellow Award and Best Doctoral Thesis Awards, they are quite prestigious. As you know, that the entire screening based on the nominations was carried out meticulously by the screening committee under the chairmanship of very distinguished scientists. And the screening committee, they submitted their report. And as per the report, the approvals were made by the competent authorities. Getting into the merit is quite important, but I would urge those who could not find a place during 2026 Fellow Awards or Best Doctoral Thesis Award, there is no point of getting disheartened. But we all should understand that it was absolutely a merit-based exercise. And during this exercise, sometimes, despite very good contribution in the field or area, we do not succeed. And much better candidates are ahead. Therefore, they should try the next year or for the other awards or young scientist awards, different categories of young scientist award and senior scientist categories as per their requirement, as per their eligibility. So with these words, once again, I congratulate on behalf of myself and on behalf of the Society for Plant Research, the entire executive body of Society for Plant Research, and wish that all the fellows who will be presenting their lectures today and tomorrow, and the candidates who have been selected for Best Doctoral Thesis Award, they will be very actively contributing towards the growth of this society and the journal. As you know, that the younger generation, they have to take lead and should come forward for the betterment and for better establishment of the society. With these words, I once again congratulate everyone present over here as an awardee, and I welcome the scientists from our executive council and from editorial board, other fellows who are with us in an online version of this meeting. So congratulations once again, and I hand it over to Dr. Jyoti for proceedings. Thank you very much.
May I now take the privilege of inviting Dr. Dolly Wataldhar ma'am, Chairman, Screening Committee SPR, to kindly deliver the introductory remark for this prestigious lecture series. Ma'am, we are honored by your gracious presence and request you to address the gathering. Ma'am?
Dr. Jyoti, you kindly check whether she is online or not.
Yes.
Just check.
Sir, I think she is not here. So, sir, should we start with calling the names of the presentees for the awards? Yes. Sir, your voice is not audible.
I think we can start the first lecture. By that time, Professor Dolly Wataldhar has got some problem is connecting. As soon as she will be here, she will address.
Okay, sir. So now I would like to call upon Dr. Rajiv Rai Chaudhary, sir, Ekrisiad, Pantacheru, Hyderabad. Sir, please, you can continue with your presentation.
Sure. Am I audible to all?
Yes, sir. Yes, sir, you are audible.
I'm just sharing my screen.
Okay, sir.
Can you see the PPT here?
Yes,
Yes, sir.
but kindly keep it in a full screen mode.
Yeah, I have kept it in a full screen. It's fine now.
Sir, I think there is some issues. Picture in picture is open.
It's fine now.
Yes, sir. Now it's visible, sir.
Okay. So first of all, I am very much thankful to Professor Bhatnagar sir and other committee members and the screening committee members for selecting us for this SPR Fellow Award of this year. And this is a actually great inspiration, as well as it comes with the great responsibilities for to do more good works, more and also the academic excellence. And also I am convey my congratulations to the other fellow members and the best PhD thesis awardees as well. So I am going to start my fellow SPR Fellow Lecture Awards. So you can see here, this is my the career and the academic roadmap. So it started from the BSc and the MSc when I have started in botany. And because I have a keen interest to study and explore more in the plant science. And after that, I have achieved my PhD in the plant biotechnology, and there I got more opportunities to involve the biotechnological tools and technologies which is associated with the plants. And then I have achieved the CSI Research Associate Postdoctoral Fellowship within in India. And then I have joined as a postdoctoral visiting scientist in Israel. So therefore, around 6 years I have been there. First, I was in the Agriculture Research Associations, and then I moved to another positions in University of Haifa. And then I again came to the ERO Volcanic Center. And after that, in 2024, I have joined as a research associate scientist in the ICRISAT Patancheru. So basically, based on all these academic uh, careers, so this is my the research goals and research pipelines, whichever I have started from the crop genetics and breeding since my master's dissertations, and whatever I am now dealing with the trait discovery and the characterizations, especially in the legumes and the other crops as well, to investigate their genetics, genomics, and the pre-breeding aspects. So first ever, I actually started my career, the research career, uh, during my master's, whenever I have worked on the mutation breeding in the dianthus, which is the most popular floriculture crops. And then I entered my PhD and I started to work on the rice abiotic stresses, majorly the heat, drought, and the salinity. And then in my first postdoctoral experiences, I have an opportunity to work on the rice metalloid stress tolerance, is specifically on the arsenic. And whenever I went to the Israel for my overseas postdoctoral program, I started on the wheat genetics and breeding. Then it's carried forward to the powdery mildew resistant genomics for the wheat. And then again, to transfer to the plant virologies, mainly I worked on the clostridovirus pathogenicity and different pathogenicity gene in those viruses in Israel. And after that, whenever I came back to India, so there is a major need to develop the Indian pulses. So I have started to work here in the pulses, majorly in the PGNP and the groundnut. And this is the translatory research roadmaps, whichever I have going through that. And this is in brief my research and the publication output. So far, I have published the 59 peer-reviewed articles with 13 book chapters, 3 edited books, and the 2 solo books or the monographs. And I have achieved till yet over 6,000 citations with 35 H index. So in national level, regional level, and also toward the world. So this is my the scientific contribution. So here I have fortunate enough to be a part to clone the PM69 gene which is came from the Waldemar wheat, and I have investigated. This is in the Israel, and this is responsible for the powdery mildew disease resistance, and also it's associated with the ROS and the cell death, how it is included in the mechanism of the wheat powdery mildew pathosystem for the disease resistance. And in Israel, I have also a part to characterize and they evaluate the strawberry and the blueberry varieties mainly, which is very difficult to grow, and in respect of their quality and also different flavors in the arid and the semi-arid Mediterranean environment. And also I have worked on the pre-anthesis flower development in the wheat in the Mediterranean semi-arid environment in the Israel, and it is mostly like to improve the wheat yield by the number of the grain setting in the spike, and also to improve the each grain weight as well. And also I was fortunate enough to work on a project that is based on the West Bengal rice landraces to improve those landraces and use those landraces in the pre-breeding way to improve the modern elite rice varieties in compared to their different abiotic stress tolerance response, their agromorphological traits, and also to improve their basic abiotic stress tolerance and the qualities as well. And I was also involved in the mutation breeding of the dianthus. The slides are not moving.
Slides are not
Yeah.
moving, yeah. The slides are not moving.
Dr. Rajiv, your slides are not moving. Actually, it is stuck on first slide.
Oh, I'm just sharing it again. Now you can see the current slides.
Yes, now it is visible.
Okay, okay, thank you. So, and later on also in the 6 number point I have mentioned here, so I involved in the improvement of the floriculture crop dianthus using the chemical mutagens, and which is very important for the export qualities to maintain our floriculture crops. Now slides are moving now. I've changed to the next one.
Yes, sir.
Okay, so that's working fine. So this is my basic translatory roadmaps that how we can use the genomics and those genetic resources. As you know, that is very much important for our nations. And also in the gene banks, we have a lot of the crop genetic resources. So we have to utilize those very precisely. And we can use also our genomic resources very precisely, and through this way some of the main achievements we can gain in that aspect, that there is we can minimize the duplications in the plant genetic resources, which is also present in different gene banks. We can help it can help us to find the candidate genes, and also we can develop marker which can transfer to the breeding teams, and they can assess in different populations and help in the marker estate selections, and also the backcrossing. And another thing is most important is the genomic selection and the prediction for specific traits in varied ranges of the crops. And as you know, this is the era of the artificial intelligence, AI. So we are also using those AIs for the very precise way of the genomic selection and the predictions as well. So here I just wanted to show you that how this is how we want to see the future agriculture and the crop development in not only in the world, also in our nation, in India. So it starts from basically from the data collections, and in the precision agriculture, it's most important to collect the data and to document them very well. And we have to analyze those very precisely and making those some decision making points, which is responsible for the resource applications and also their evaluation, and that's leading to different data sensing points. So in this way we can utilize the AI in our current research So different aspect wherever we are working on that. And this is summarizing some of my recent achievements of the projects. That's two projects I have was funded as a principal investigator, and it was two projects that I was in Hope PI, and it was funded earlier. So recently I got one sanctioned projects on the PGNP annuality and the photothermosensitivity from the NRF Early Career Grant. And also recently we have completed one project from the DBT 2 years, which is based on the haplotype mapping in the PGNP. And earlier in the University of Haifa, Israel, there was one project which is based on the wheat powdery mildew disease resistant genomics, and another one wheat pre-anthesis and the early vigor, which was also completed for the 2 years from Israel. So this is the award list that I have gained till yet, and in this year I got 2 awards: Pioneer Research Scientist Award from Anjania University, and another Best Poster Award from the BHU. And this is the third one that I have gained now with the SPR Fellow Award for this year. And also 2023 I was awarded Young Researcher Award from ISHARD conference. And also earlier in 2019, I got the Travel Award from the International Wheat Initiative Society from Germany. And I have attended their International Wheat Congress, first International Wheat Congress in Canada. And also 2013, I got the Best Poster Award from Vishwabharati Central University. And 2012, I was selected as a fellow member of the Linnean Society in London. So this is the like a brief showcase for the editorial assignments. So with from 2022 now, I am continuing as the editorial board member of the South African Journal of Botany. And also from 2025, I was associated with the CAVI reviews and the BMC Plant Biology and the Plant Biotechnology. And last but not the least, I was also associated in the last year with Vegetos for the section editor of plant physiology and the biochemistry. And here more than 10 journals where I am now participated for the reviewing of various articles till yet. So this is the last slides of my presentations, and here I am going to acknowledge my collaborators, Dr. Roy Ben David, Professor Zion Fahima, and Professor Munir Mawasi from Israel. From ICRISA, there is Dr. Manish Pandey, and also one of my colleagues and the collaborator, Dr. Yinghui Li, is from China, Dr. Ajay Kumar from
Thank you very much, Dr. Raji, for your wonderful presentation. Over to Dr. Jyoti.
So thank you for your presentation. Now I would like to call Graphic Era Deemed University, Uttarakhand. Hello,
Check the connectivity issue.
am I audible?
Check the connectivity issues there. Connectivity issue.
I'm trying to fix it.
Yeah.
Can,
Dr. Rupak is having some connectivity issue.
So I think it is from the office of Vegetos.
so, sir, can we call upon the next candidate if there?
Yes, I think so.
Yeah, Dr. Jyoti, if can you adjust me here because I am from Poland at present, so I may have some another lecture. If you can shift my lecture to here, it will be much better in this place.
Okay, Dr.
Yeah.
Shamsul Hayat.
Please, sir.
Am I audible?
Yeah.
Yeah, Dr. Jyoti. Kindly invite Dr. Shamsul Hayat. His PPTs are here with our office. They will share. Okay, Dr. Jyoti.
Thank you, sir. I'm ready, sir. Slides are in your
Please.
office.
Yeah. I've asked my office to share your slide, Professor.
Thank you, sir.
Yeah. Just a minute, please. So, Dr. Jyoti, you kindly change the sequence.
Yes, sir. So, calling Dr. Shamsul Hayat, sir.
Yeah.
University. Okay, sir. Sir, you can start with the presentation as soon as the slides will be shared by the.
Slide is coming. Till that, I'm just starting that myself is Professor Shamsul Hayat from the Department of Botany, Aligarh Muslim University, Aligarh. And at
Doctor, just a minute.
Okay.
My office has informed that the slides have not been received.
Oh, sir, I have already sent you. So, but I'm just start to share if it.
Please send it again. Abhi, you have just sent it. No issue. They are sharing.
It is received?
It has just reached. Yeah.
Okay, sir.
Just we have
Hello?
to wait for a moment.
Okay. Thank you.
Hello, am I audible?
Yes.
Yeah.
Yes.
Dr. Rajiv, you kindly keep your mic mute so that Professor Shamsul Hayat may start his lecture.
Switch it on.
Yes. I can start, sir?
Yeah, sure, Professor Hayat.
Yeah. Myself is Professor Shamsul Hayat from the Aligarh Muslim University, Aligarh. At present, I'm visiting professor at the University of Białystok, Poland. So I'm just giving my presentation from here. Next slide, please. So this is my academic profile. Just I'm professor since 2012 and about 34 years of research experience and 20 plus years of teaching experience. And I have expertise in the plant physiology and structural biology. Next, please. So till now, this is my significant contribution. We are just the pioneering work on the breast stars in India. The first group who started working on the breast strand is from the Professor Sita Ram Rao in Hyderabad University. And next, we have just one. And I have also started my working on the salicylic acid signaling under stress. And besides that, I have also worked in the hormonal crosstalk in the plant science. So at present, my work is related to that.
Dr. Hayat, your voice is not clear. Maybe the connection issue or network issue.
At just a minute, sir. I'm just completing. At present, my area of research is nanoparticle mediated stress tolerance. That is clear?
Yes, it is okay now.
No.
Yes,
Please,
sir.
next slide, please. So my research vision is that how the plant hormone, especially the breast steroids and salicylic acid, ameliorate the stress. Besides that, the synthesis, characterization, and application of nanomaterials on plant. So, but it is not possible to present all the work. So I am just presenting few findings here. Next, please. So it is related to that of the breast steroids. Next, please. Yes. Breast steroids is a class of a steroidal plant hormone that play an essential role in plant growth and development and implicated in wide range of morphological, physiological, and biochemical responses in plants. They have ability to confer tolerance against various abiotic and biotic stresses. Next, please. So actually it has a variety of role in the jets. It improved the lycopene content, control the morphological and the tertiary element, activated the cell cycle, as well as the modulation of the apoptosis. And overall, the main thing is that it has increased the seed yield in the various crops from 10 to 17% till now. Next, please. So this is one of the experiment where we have applied the one of the brasinosteride that is called as the epibrasinoide as a foliar spray on the tomato crop. And here it has been reported that the shoot length as well as that of the root length at the, both the stage of the growth has been increased by the application of the brasinosteride, but the 10 to the minus 8 molar is proved to the best among them all. Next, please. So just you can see it clearly from the pot experiment where the 10 to the minus 8 molar has increased the overall growth. Next, please. So here, this is the one of the most important parameter, that is the net photosynthetic rate, that has been also increased by the 10 to the minus 8 molar of the epibrasinoide, and the same has been happened with that of the stomatal conductance. Next, please. So again, the internal carbon dioxide concentration and transpiration give a same trend with that of the 10 to the minus 8 molar concentration. Next, please. So overall, the lycopene content, beta carotene, as well as the fruit yield. That is, our aim was to increase the actually the fruit yield, and we got a success that the 10 to the minus 8 molar has been able to increase the seed yield in the case of the tomato plant. Yes, next, please. From this experiment, we have just concluded that 10 to the minus 8 molar of epibrasinoide positively increased the photosynthesis and ultimately the yield of the tomato crop. Yes, next, please. So our next aim is just related to the phyto-assisted synthesis and characterization of nanoparticle and their effects on the performance of Brassica gentiana. Next, please. Nanotechnology basically involves manipulating a matter at the nanoscale, typically between 1 to 100 nanomole, where materials gain unique properties due to their size, shape, and surface characterization. At the nanoscale, particles can interact directly with the plant cells, enabling precise delivery nutrients and agrochemical into plants. Additionally, we can say that the nanomaterial exhibit a high surface area to volume ratio, which enhances their reactivity, absorption capacity, and efficiently in improving the plant growth, stress, as well as the nutrient use efficiency. Next, please. This is the nanoparticle that belongs to 1 to 100 nanomole. Then it has a zero dimensional. It has a van der Waals forces, nanocatalysis, as well as that of the high surface to volume ratio. Next, please. The phytosynthesis, that is the green synthesis of the nanoparticle, they are basically eco-friendly, have a non-toxic, most economical, as well as that have a sustainable development. Next, please. So we have just analyzed the cerium oxide nanoparticle, that basically it is exists in 2 ionic states and has the high abundance among the rare earth element. Yes, next, please. So actually, the application of the cerium oxide nanoparticle has been in the various fields. They can be act as enzyme catalysis. They are also used as anti-cancer. Then drug analysis. They have antioxidant capacity. They are antibacterial. They also have a phytocatalysis in very much used in the medicine as well as the delivery system. Next, please. We have selected a crop that is mostly in our area is used, that is the Brassica gentiana. It is an annual herb, as you know that it has been. It is not going to be in detail. Very much we are known about you. Next, please. So my objective is that first to synthesize a cerium oxide nanoparticle from a graded crystal size from the leaf extract of Moringa oleifera. Then to perform the characterization of the synthesized nanoparticle, then we have to analyze its individual effect of the selected concentration of the three different sizes through the foliar application. So to analyze the interactive effect of best concentration of each of 3 nanoparticle, which we have just done it, and to analyze their interactive effect. Next, please. So this experiment conducted with an aim to synthesize cerium oxide. Next, please. This is the basic process, that is the flow chart we have just used. This is one of the, we have just used the leaf powder, and this is the overall the process which has been just here. With going to the different process system, we are just analyzing its synthesis. Yes, next please. Then we have done XRD, that is the crystal size and symmetry can be done, FTR, functional group analysis, DLS and ZP, that is size and surface charge can be done. And finally, the size and shape has been analyzed using the SEM and TEM techniques. Next, please. This is the result that shows the 3 different XRD pattern as well as that of the FTR pattern in all the 3 chemicals. Yes, next. So here you can see that this is the 3 different nanoparticle. Just I named it ABC. They have a crystalline size different, and again their zeta potential is different and DLS is different. Next please. This is the SEM and TEM technique, which clearly indicate that all the 3 particles have a different size and shape. Next please. So from this, we have just concluded that successful green synthesis of nanoparticle were achieving using M. angifera leaf extract, conforming an eco-friendly, cost-effective, and sustainable approach. 3 distinct nanoparticle sizes have been synthesized, obtained by optimizing synthesis condition, demonstrating effective control over particle size as well as that of their morphology. Yes, next please. Then we have just done a pot experiment with all the 3 concentration. Next, please. So from this figure, you can clearly indicate that all the 3 concentration as well as that of the 3 different type of the nanoparticle have a significant impact on all the photosynthetic parameters, and the photosynthesis, stomatal conduction, and transpiration rate, and internal carbon dioxide increases by all the 3 concentration and all the 3 nanoparticle. Next, please. This slide clearly indicates that all the 3 nanoparticles have a histometal localization of superoxide anion, and at the 45 days after sowing, and all the 3 have a different type of localization. Yes, next please. So from here you can say that histochemical detection of lipid peroxidation was also done in the leaf as well as the root, and just see that from the control, and these 3 different nanoparticles have a different histochemical detection in the case of the leaf as well as that of the root. Next please. So here we have a confocal microscopic image of root showing that is the ROS localization as well as the cell ability, and all these 3 nanoparticles have a different color. Next please. So from this you can say that the Fourier mode has also ultimately increased the yield parameter, that is the number of seed, number of pods, and all the 3 nanoparticle and their concentration has the capability to increase the overall yield content. Next please. So our next just step is that how the mitigation can of the heavy metal by using these synthesized nanoparticle can be possible or not. Yes, next please. So in this case, we have just developed a novel strategy to mitigate the copper-induced oxidative stress in tomato. And here we have just used the green synthesized nanoparticle that is from the Phoenix dactylifera. In the earlier experiment, we have just used mangifera, but here we have just used the date. Yes, next please. We performed integrated analysis of physiological responses, biochemical changes, antioxidant defense system, provided a clear mechanistic insight into the redox homeostasis regulation as well as that the stress mitigation by the cerium oxide nanoparticle. Yes, next please. So here I'm just coming only on the images of the scanning electron microscope and TEM, because all the other procedure which are almost similar, I have just described in the earlier part. So you can see that they have a different size as well as that other different shape of the nanoparticle that obtained from the leaf extract of the founding Dactyliflora. Here, one thing which I want to tell you very clear, that the size obtained from the Phoenix Dactyliflora is comparatively better than size obtained from the Mangifera. Yes, next please. So here you have a clear-cut picture that in the control plant, the stomatal pore is as comparatively less as compared to the other. When they are applied at the even in the stress condition, they are going to be decreased, but when they are going to apply in association with the nanoparticle, the overall the stomatal pore can be increased. Yes, next please. So from here you can say that the effect of different level of soil applied copper and obligate different concentration of the nanoparticle synthesized applied through foliar mode on the leaf area as well as the chlorophyll, photosynthetic and stomatal conductance. And this figure clearly indicates that the stress generated by the copper can be partially overcome by the application of the nanoparticle. Yes, next please. So again, we have just hydrogen oxide content and with that of the in the copper as well as that of the nanoparticle treated plant, and we can just clearly indicate that the stress generated by the copper can be negated by the application of the cerium oxide nanoparticle. Yes, next please. So here again, the superoxide anion is also shows a similar pattern as that of the earlier slide. Next please. So the MDA content is also have a almost similar pattern, but the effect generated by the copper can be negated by the application of the nanoparticle. You can see just from these three figures you have. Yes, next please. So here, this is the historical detection of the lipid peroxidation with shift reagent in the roots of lycopersicon that has been treated with that of the copper as well as that of the cerium oxide nanoparticle. So more intense pink color that indicates the more lipid peroxidation in the cerium oxide treated plants. Yes, next please. So this is a confocal microscopic images which shows a ROS localization, and you just see from this figure there's abundance of fluorescence substance that is green in the color, indicate more ROS accumulation in the roots of lycopersicum that is treated with the copper, and obligate is spread with that of the cerium oxide nanoparticle at 45 days after treatment. Yes, next please. This figure also shows the cell viability abundance. Here, the treated with the copper as well as that of the cerium oxide nanoparticle have a different trends. Yeah. Next, please. So finally, we have just concluded from this experiment that of the different concentration that is of all three applied through Fourier mode, 100 ppm of the cerium oxide nanoparticle with the particular size that is for A, that is 4.5 nanometer, proved best to enhance all the growth, photosynthetic and biochemical parameters. Overall, the study clearly indicate that the mediated synthesis as a reliable method for prolonged functionally different efficient and to enable cerium oxide nanoparticle and their successful application in the mustard plant. Importantly, the finding highlight a clear size dependent effect where nanoparticles of a smaller size exhibited the most pronounced positive impact on the growth and performance of mustard plant, demonstrating their superiority efficacy than that of the larger particles. Yes, next please. Yes, I'm going to finish. This is the award, and I'm thankful to the society for the award of the fellow. Then I have the fellow of Linnean Society, Indian Society, Nath Associate, Bhaskar Fellow, INSA Visiting Fellow, Hiranal Chakravarti Award, Khankoje Gold Medal, Mass Young Scientist, and Visiting Professor. Next, please. This is the research output that I have guided 12 PhD students, supervised 30 MSc students, 2 MPhil students, international cooperation with Poland, Japan, Turkey, and Saudi Arabia. More than 200 publications, 35 book chapters, 18 edited books, A citation of more than 30,000 and H index of 80. Yes, next please. This is my 12 student who have completed the PhD under me. Yes, next please. So this is the at present the Google citation is here. Next please. Thank you so much, sir, for giving me opportunity to present my work. But it is not that all work cannot be presented in a very short time. Thank you, Bhatnagar sir, for giving me an opportunity.
Thank you, Professor Shamsulhayar, for wonderful presentation. Despite your very hectic schedule as visiting professor in Poland, and I wish that your stay will be very comfortable and very fruitful for the scientific community. Thank you
So
very
thank you,
much,
sir.
Professor Shamsulhayar.
Thank you, sir. Just I'm leaving. Thank you so much.
Thank you.
Thank you, sir, for your presentation. So, now we can call upon Dr. Sangeeta Bansal, ma'am, ICAR, National Bureau of Plant Genetic Resource. Ma'am, over to you.
Thank you, Dr. Jyoti. Good afternoon, everyone. I hope I am audible.
Yes, ma'am.
Slides are visible.
Yes, ma'am.
So good afternoon, everyone. First of all, I would like to thank Society for Plant Research, especially Professor S.K. Bhatnagar, sir, and the entire screening committee, as well as the chairperson of the committee, Dr. Dolly Wataldhar, for conferring this prestigious recognition upon me. Thank you so much. I am Sangeeta Bansal. I am presently working as principal scientist in the Division of Genomic Resources at ICAR National Bureau of Plant Genetic Resources, New Delhi. Our institute hosts the world's second largest gene bank. So moving on to my professional experience, I have received my PhD degree from GB Pant University in molecular biology and biotechnology. I started my career as assistant professor at the Sardar Vallabhbhai Patel University of Agriculture and Technology, Meerut. Then I joined ICAR CFET Ludhiana in the year 2011 as senior scientist. Thereafter, I got transferred to ICAR National Bureau of Plant Genetic Resources in the year 2017 as principal scientist, and I am continuing at ICAR NBPGR New Delhi. So during my career at 3 different organizations, at Sardar Patel University, I was basically involved in teaching of UG and PG courses. And since we were the first faculty that have joined the university, so we were extensively involved in the designing of course curricula of BTech and MTech Biotech courses for the Department of Commercial Biotechnology there. At ICAR CFET Ludhiana, my research basically focused on the various aspects of food biotechnology that included process and product development and detection of adulterants and contaminants of different food products through molecular methods. At ICAR NBPGR New Delhi, that generally focuses on research related to plant genetic resources, characterization and conservation. I have worked upon in vitro conservation and cryopreservation of plant genetic resources, as well as development of genomic resources of important crop plants. In my presentation, I will try to touch upon few significant research achievements. So these are the different projects which I have handled, and I have handled around 7 externally funded projects and more than 10 institutional projects. This table enlists the different externally funded projects that I have handled. So I will be starting with my work at ICAR CFET Ludhiana. As I have told, I was involved in the development of different processes and products. So in that, I was involved in the development of non-dairy based probiotic foods. For this work started with the isolation and characterization of lactic acid bacteria from different fermented products. And we could be able to successfully isolate some putative lactic acid bacterial strains whose sequences were submitted and registered at NCBI Gene Bank with accession number mentioned in this table. This work has led to the publication in the Journal of Vegetables itself. Once we have isolated the lactic acid bacteria, we went for the characterization of these bacteria for putative probiotic potential. And after that, we could be successfully able to isolate about 7 putative probiotic isolates. And these probiotic isolates were successfully deposited to different depositories. As you can see in the table, these 3 isolates were deposited in the International Depository at CSIR Imtech, Ludhiana. And these 2 were deposited at NBAIMO, that is National Depository. Once we have the probiotic isolates with us, then we went for the development of different non-dairy products. The objective was to provide dairy alternative to the lactose intolerant population of the country. As we all know that milk contains lactose, sugar, and a substantial proportion of our country, substantial proportion of the population of our country is sensitive or lactose intolerant. For those, we have tried to develop some dairy alternatives. So we have developed non-dairy based probiotic soy yogurt. We have also developed peanut probiotic yogurt. And both these technologies were successfully published in very high impact factor journals. Then I have also worked upon development of a microbial method for the production of protein isolate or concentrate from oilseed cake and meals. India is the largest producer of oilseed cakes. It produces around 17 million tons of oilseed cakes or meals annually. And then this cake is exported to other countries where it is converted into the production of protein isolate and then imported to the country. So we have tried to develop the process for production of protein isolate or concentrates from these oilseed cakes and meals. Traditionally, protein isolate is prepared by using alkaline extraction followed by acid precipitation. So we have tried to omit the use of these chemicals by using microbe in place of concentrated HCl. The strains which have been isolated in our lab, that is Lactobacillus fermentum DB4, that is also having probiotic potential in association with Lactobacillus acidophilus was used in this process. And with the help of this process, we could be able to successfully develop a process protocol for production of protein isolate that contains around 90% of the protein from this oilseed cake or meal. This research has led to granting of one international and one national patent. This technology has been patented in US as well as India. And this technology has been successfully transferred to different food processing industries from the year 2021, and still there are requests for licensing of this technology. The next aspect of my research was development of molecular methods for detection of adulterants and contaminants. In this, we have developed a molecular method for detection of aflatoxin producing fungi in groundnut and rice. And we have targeted the genes, those are involved in aflatoxin biosynthetic pathway. We have tried to develop specific primers against these genes. And we were able to successfully develop a method for successful detection of aflatoxin producing fungi specifically. In addition to this, we have also worked upon detection of different adulterants of basically biological adulterants in different high value products using molecular methods like papaya adulteration in black pepper, safed jeera adulteration in black jeera, and safflower or calendula adulteration in saffron. So in this, we have developed either specific primer like star markers, or we have developed DNA barcode-based markers that could successfully detect even 1% of adulteration of these adulterant in other important crops. This research has led to the development of around 25 sequences, and these sequences are submitted and registered with NCBI Gene Bank. And this research has also led to publication of papers in high impact journals. And we have also come up with monographs for detection of these adulterants and contaminants of different adulterants in these important crops like black pepper, saffron, and kala jeera in the form of monographs. At ICR and BPGR, as I have told, that I am basically involved in vitro conservation and cryopreservation of crop plants. And under in vitro conservation, I was involved in the conservation of tuber crops that contains around 526 accessions, and spice and industrial crops that contains 227 accessions. In addition to management and conservation of these germplasm, we also work upon research aspects, and we were able to develop different in vitro propagation and cryopreservation protocol. We have developed the protocol for in vitro micropropagation of sweet potato. We have also developed protocol for in vitro multiplication of Xanthosoma sagitifolium. In addition to this, we have also worked extensively on the industrially important crop Rubia cordifolia, that is also known as Manjistha. And we were able to develop an efficient direct organogenesis protocol for micropropagation of this Rubia cordifolia. In addition to in vitro conservation, I was also involved in cryopreservation of different plant germplasm. Cryopreservation is a process for long-term conservation of plant genetic resources under liquid nitrogen at the temperature of -196 degrees centigrade. And this is the method for long-term conservation of vegetatively propagated crop plants or non-orthodox seed species. So we have developed different cryopreservation protocol. Like in this slide, you can see the example of Rubia cordifolia itself. These are some other important publication or some other important research accomplishments that are related to in vitro multiplication and cryopreservation of different plants. Basically, these involve wild species of ginger, wild species of piper, and jojoba. In addition to in vitro conservation, I was also involved with the management and maintenance of non-orthodox plant genetic resources in National Cryogene Bank. And I was involved with the management and maintenance of Cryogene Bank till 2024. And till that time, the total holding of the National Cryogene Bank was 14,130 accessions of different non-orthodox seed species. During that period, we have also collected the germplasm for Bunim persicum or Kala Jeera from different regions of Western Himalayas, and these are successfully conserved in the National Cryogene Bank of our institute. At the moment, I am posted at Division of Genomic Resources, that is basically assigned with the duty of development of genomic resources and molecular characterization of different plant genetic resources. So in this aspect, I have worked upon the development of genomic resources for Kala Jeera or Bunim persicum. Before our work, there was no report of any sequence information for Bunim persicum or the entire Bunim genera as well. We were the pioneers who have gone for de novo genome sequencing of Bunim persicum and led to the development of genomic as well as EST-SSR for this important crop. Again, this work was published in a very high impact journal. With the help of the genomic resources that were developed during this project, we have also generated a database that is openly accessible at our website. You can access the genomic information or genomic markers for Bunium persicum with the help of this website link from anywhere in the world. And this database is also copyrighted in the year 2025. So these were the important research accomplishment I have tried to touch upon, just a significant research accomplishment of mine. In addition to this, I have been involved in various administrative roles, like I have worked as in charge of PMB cells, vigilance officer, transparency officer, warden of the hostel at Sardar Patel University, and in addition to this, many more publication in form of books, technical manuals, and popular article, research papers that have been done. And with this, I would like to acknowledge the ICR Director, ICR MBBJ New Delhi, Director ICR CFET Ludhiana, and Vice Chancellor Sardar Vallabhbhai Patel University of Agriculture and Technology Meerut for all the resources and facilities that has led to these research accomplishments. Thank you very much.
Thank you so much, ma'am, for your presentation. Now we can call upon Dr. Hukum Singh, sir, from ICFRE Forest Research Institute, Uttarakhand. Sir, over to you. Dr. Hukum Singh, sir. Sir, he's not here in the attendees.
You can move forward. If he appears,
Okay, sir.
he'll present.
Okay, sir. So now we can call upon Dr. Ingudam Bhupe Chandra, sir, from Central Agriculture University, Imphal, Manipur. Sir.
Dr. Bhupen Chandra,
Yes, sir, I'm on the line.
yeah, please continue.
Okay, please. Okay. So. Good afternoon to all. Am I audible, sir?
Yeah, you
Am I
are
audible?
Yeah.
Okay, good afternoon to all. Respected Chief Patron, Professor R. Hanchidalji, Founder, Chairman, Professor S.K. Bhatnakarji, President, Professor Banaraboji, and Moderator Jyoti Singhji, and to all the achievers of the Fellow Awards. So first of all, I extend my deepest sense of gratitude to each of you for conferring me the prestigious SPR Award 2026. So I'm truly honored by this recognition, and I'm really appreciating you all for advancing this agricultural research. So now I start my presentation. Right. So I start directly with the salient achievements. So as of now, I'm holding the post of Deputy Director and affiliated to the Director of Extension Education, Central Agriculture University in Imphal. And the core responsibilities included technology dissemination, program execution, capacity building, and advisory feedback. And formerly, I was associated as an associate professor in soil science and agriculture chemistry in Central Agriculture University in Imphal. So the present work profile and areas of expertise. My expertise is in the field of soil science and plant nutrition, integrated nutrient management, carbon sequestration, agricultural extension, that is related with this on farm trial, frontline demonstration program, technology dissemination, and farmers capacity building. That is directly relevant to the area of my directorate. Apart from that, I work on soil chemistry and soil analysis. There is characterization of heavy metals, assessment of soil. Then sustainable and organic farming, then climate smart agriculture. These are some of the publications. So in total, I have 158 publications, and I published in some of the high NAS rated journal from Elsevier, Willie, Springer, and Taylor Francis. And I have around 3 training manuals, 6 number of published edited books, and 45 numbers of book chapters. So this is my research profile, and these are the technologies which we have developed since the inception of my research work. So I've published in some of my developed technologies in high NAS rating journal in more than NAS rating 10. This is some of my ongoing project which has been completed, and it is under TSP tribal subplan, which is in the tribal inhabited area of Tamengung district. It's one of the remotest area. Its district is located in Manipur. So it was one of the most aspirational district that needs development. So under the guidance of our center, our director, our ICR, it has been developed. And these are the patent. This smart irrigation is using the real-time kinematics GPH. And some of these are the awards which I have got it. These are Distinguished Scientist Award, then Young Scientist Award from Scientific Development and Agriculture Technology from UP. And these are some of the accomplished projects. So there are some issues. So I've handled 2 projects as a co-PI and a PI, which cost 2.13 crore and 1.5 crore. So coming to one of the topics which I have to one of the project, and this is the research which I have conducted. So this the title is the elucidating the synergistic effect of this farmyard manure along with the boron. This boron is one of the micronutrients that is required in minute doses, which when incorporated with the farmyard manure, means organic manure, it has given a good feedback from the farmers. So this research gap include this current studies. There's a dirt. That means there's lack of comprehensive insight into interaction between boron and enriched farmyard manure in the soil health, in nutrient dynamics, and crop productivity, particularly regarding the evidence-based optimization of this application rates for sustainable agriculture and management. So now we have to focus more on this efficient use of this micronutrient along with the organic manures. So when these two, this micronutrient and farmyard manure, when we apply, there's a synergistic effect. So this synergistic effect, it has the potential to improve the soil health as well as the yield of the crops. So the experiment was done from 2015 to 2017 in Jorhat district under the Assam Agricultural University. So for this, we have used this multivariate data analysis for assessing all the parameters which I have taken. Now, after the application of this boron and the farmyard manure, so what we have done is that we have collected all the soil sample and we have analyzed in the laboratory along with the yield. And apart from that, some of the most important parameters like the Sanon diversity index, the essential yield index, which assess the microbial carbon sources, we have studied. And after the detailed study, we have drawn a graph, and this graph, this indicates that this highest dose, this farm boron at 1.5 tons per hectare in combination with the 15 tons per hectare, this farmyard manure, it accomplishes the highest the French bean pot yield. This French bean pot yield, it corresponds to this fresh pot yield. Okay, so the combination of 15 and 1.5 kgs per hectare boron, it accomplishes the highest yield. So like that, this GME and sustainable yield index, Sanon diversity index, these follow the same trend. So the highest of these parameters, this sustainable yield index, the Sanon diversity index, they are all corresponded with the highest doses of boron and the highest doses of enriched farmyard manure. So now coming to the soil quality index. The soil quality index is calculated to figure out which where we have to focus on. This soil health, it encompasses this physical, chemical, and biological properties. So let's take example when we talk of the soil physical parameters. This bulk density is one of the most important parameters. So like that, in case of soil chemistry and soil quality parameters, then we should take the CEC, cation exchange capacity, then pH, then the nutrient like macronutrient and nitrogen, phosphorus, potassium. These are all taken into account. And apart from the biological sources, we are more focused on the soil enzymes and soil microbes, like soil fungi, soil bacteria, soil actinomycetes. So we have used this hierarchical cluster analysis of the soil health indicators. Here, the soil health in this cluster analysis indicates that there is a clustering of the most important parameters. Like here, we can see this available nitrogen, which is in close proximity with MBM, microbial biomass nitrogen, then soil respiration rates, along with bulk density. So these 4 or 5 parameters, these are very much important parameters, and this indicates that these parameters have directly linked with the soil health. So in order to validate the results, we have to use this network and correlation plot. So integrating with the Pearson correlation matrix. So here we can see this soil quality index SQI that correspond directly here, this pink line. This pink line, it corresponds with the available this nitrogen. Here we can see nitrogen, then available phosphorus, along with important micronutrients like iron, copper, manganese, zinc, and all. So this validates that these parameters are directly linked with improving the soil health. Now we are coming to the principal component analysis of all the important soil parameters. So this will reduce the dimension of the most important parameters. So here, after analyzing the soil parameters using this principal component analysis, here in the eigenvector, this CEC, this cation exchange capacity, then soil organic carbon, available nitrogen, available phosphorus. These parameters, these are having the highest loading. It means that it is directly effect on the soil health and then crop yield. Because these parameters, as we know that this nitrogen is one of the most important parameters that is directly linked with the crop yield. And this potassium, phosphorus also, there is directly linked with improving the roots architecture. And in order to validate the results of the other finding, we have used this artificial neural network and machine learning. So here we can see the hidden layer. That's in hidden layer, this available nitrogen, soil organic carbon, then exchangeable calcium, this and multiple micronutrients. These are all directly linked with improving the yield of the crops. So this is one. And after the work is, after the accomplishment of the work, we have submitted or published to some of Elsevier journal, and it was published in the subsequent years. So the conclusion, this study demonstrates that integrating this optimized boron and enriched farmyard manure significantly enhance the frenzied productivity and sustainable yield. And this multivariate analysis, as we have shown in 3 or 4 slides, confirms that the highest level of boron at 1.5 kg per hectare and enriched farmyard manure 15 is the most optimal dose to elevate the soil quality index and reduce acidity. So with this word, I wrap up my presentation. Sorry.
Okay, thank you, sir, for your presentation. Now, as Dr. Hukum Singh sir is available, so now we would like to call upon Dr. Hukum Singh sir, ICFRI, Forest Research Institute, Uttarakhand. Dr. Hukum Singh sir.
Yes, I am present.
Okay, sir.
Is this visible?
Yes, sir.
Okay, thank you. Thank you. Thank you, sir, for giving me this opportunity to present my work, whatever achievement actually I have made over during my career. So, as per my education is concerned, I am postgraduate in botany and environmental science and environmental economics. Presently, I am pursuing MSc in environmental sustainability. So, in addition to that, I acquired my PhD in plant physiology with specializing in environmental science from GU Pant University of Agriculture and Technology. So before starting, I would be discussing something which is very globally important. You know, it is important to understand the major global climate and environmental changes that not only human or vegetation face, entire actually ecosystem or land earth system facing actually. So you can see from this figure where how actually CO2 is accumulating in our atmosphere. If you compare the present scenario of CO2 present in the atmosphere with the pre-industrial era, you can found how much CO2 has been increased. So present scenario, I think around 425 or 426 ppm CO2 is present. If you compare with this pre-industrial era, you can find 280 actually. So you can see how gradually it is increasing. And apart from that, IPCC has also projected the increment of atmospheric CO2 in the our environment. Based on different scenario, it has been projected that by end of the century, we will be having this atmospheric CO2 concentration between 900 to 1000 ppm. So it is actually very high concentration of CO2 we will be facing in the environment. So that's increased CO2 will increase the atmospheric temperature. So parallelly, the temperature has been projected with an increase of plus 1.5 to 4.5 degree by end of the century, but it is based on the different scenario which have been developed by the IPCC. Like in some scenario, if we adopting some adaptive and mitigation measure, then there will be different scenario. If we leave it the condition as it without adopting or employing the adaptation and mitigation measure, then we will be having this much quantity of CO2 and the this much temperature in the atmosphere. Apart from that, one major problem which we are facing. So most of the population who is residing in the urban area is exposed to temperature and elevated CO2. Apart from the air pollutants, so other air pollutants like SOx, noise, particulate matter are also can be found in the high elevated level in the atmosphere, and also urban heat island actually. So that ultimately gives the very severe stress to the urban environment. So our aim only is to focus on the urban vegetation or the plantation species or forestry species from which we derive different huge quantity of ecosystem services like cooling, carbon sequestration, and air pollution removal. So these are actually the main ecosystem services which we are retrieving. So all this scenario has evolved a question. Question is that how actually the do tree physiologically respond and adapt, and how actually they will contributing in the mitigating the climate change and environmental impact. So plant actually follow different adaptation and mitigation strategy to combat the environmental and changing climatic conditions. So based on that, actually we have created the facility at FRI just to understand how actually the plant will respond, whether the medicinal plant, agroforestry species, or other important non-timber or timber forestry species, including urban vegetation also. So there are different approaches which we have, which we are employing or at FRI just to understand the physiological response of the species at stand level or leaf level and landscape level and ecosystem level actually. So there is one approach you might be knowing that is known as automated open top chamber facility. Here you can actually you can create the future climatic condition like elevated CO2 temperature if you want to increase, or if you want to create the climatic condition of 2050, then you can increase the CO2, you can increase the temperature, when you increase the other greenhouse gases also, you can have high level of ozone and other air pollutant also. And apart from that, we are also adopting this same condition, but it is the natural actually area in urban area. So that can be urban different environmental gradient of urban area that can be explored as a natural climate or natural open lab for climate change or environmental related study. So in that, what actually we are doing, we are considering the different labs like residential area where you can find different, high air pollutant, high temperature, and high greenhouse gas also, and heavy metals also. Then industrial area, there will be different air pollutant or different climatic scenario, and residential area, then commercial buildings. So why actually we are doing this? Just to validate the result or the scenario or pattern of the plant response to climate change, which we are obtaining in the open top chamber condition. So the second approach gives you some similar, it's actually natural conditions. Apart from that, we are also doing the climate change vegetation modeling. So this is actually the computer-based modeling, where we can use different models or software through which we can assess the different plant phenological, physiological, or biochemical responses, like how the carbon sequestration will be in future climate, how the phenological adaptation will be there, how the life cycle of plant will be changed, and how the plant will adopt, and what will be the impact of this changed physiological process on the ecosystem process. And apart from that, one approach recently we have adopted, that is actually long-term ecological modeling, and that we have already established a permanent plot where we are continuously monitoring the response of individual plant and at ecosystem level also actually. So this will give you real-time data to understand the plant response to different climatic scenario. So this is actually the, I told you that we have constructed this facility at FRI. This is called the open top chamber facility. This facility generally have three components. There's the CO2 distribution unit, open top chamber, control room. In OTC, open top chamber, where we grow the plant, and from control room, where controller is there. From there, you can manage the amount or concentration of CO2 and level of temperature. If you want to increase the 800 ppm of CO2, just you to enter the 800 ppm level of in the controller that is connected to the respective chamber. Automatically valve will open from the CO2 distribution unit, and a desired level of CO2 will be managed. Similarly for temperature, similarly for other gases also. So in urban area, you can see here different climatic conditions in different land. Like rural, we have different; in city area, we have different. So you can assess the plant response to different climatic conditions. So this is actually something which we have contributed significantly to science. Like we assess the tree response to elevated CO2. What achievement actually we had made? So illustrated physiological and productive response of trees in the elevated CO2 and concentration. But apart from that, actually the physiological response is also governed by different interaction actually with the CO2, like nutrient, temperature. So if we want to have the positive effect of CO2, that is called the positive CO2 fertilization. So we should have the proper availability of nutrient and and other also like water also, and how other soil microbes like PGPR and heavy metal interacts with that. So all complicated process determine the adaptive resilience of the particular species. So what observation we have got? We reported under the elevated CO2 that photosynthesis in carbohydrate production and carbon capturing capacity was increased. That will lead to induce or increase the biomass productivity, water efficiency also increase. But what we reported, continuous exposure of CO2 may lead to the decline the soil nutrient availability. So there may be a constant for growth and development. So in that condition, CO2 may not have significant positive impact on the plant. So what actually we do? We also require nitrogen addition. Similarly, in case of agriculture, which we are doing the practices. But in case of agroforestry species or tree species, there is no recommended dose of nutrient fertilizer. So we have also optimized the nitrogen level. So which quantity of nitrogen is required for proper growth and development of plant or productivity under the elevated CO2 concentration. Similarly, we use the PGPR just to enhance the CO2 fertigation effect. And what we reported, apart from that, we have also reported that we found species-specific adaptation mechanism. That is, of course, the species have different adapting mechanism. So you can see from this picture, we exposed this is one of the Rubilia robusta species. If we exposed to different limit CO2 concentration, what threshold we have reported? So 800 CO2 concentration, this maximum carbon assimilation was achieved with the biomass, better biomass production. If you exceed beyond the 800, there you can see from there decline in carbon assimilation, but it is higher than the ambient. So we can say that this particular species may survive in this condition only actually. So we can recommend this species to urban area or the area where, the industrial area where high CO2 concentration is there, that can be planted so that maximum carbon can be removed from the atmosphere. And apart from that, we have also calculated the cooling impact, how much cooling can be created by it, because this is actually the major question in forestry species. Most of the time people ask which species can create more cooling. So I will come to the next slide. This is another species, Acacia auriculiformis. So what the result actually we found. So 30 micromole per meter square per second we reported this CO2. It is very high in case of agroforestry plantation species, but it is reported in rice over the 30 pp, even up to 43 micromole CO2 is being captured by the rice also. But in case of tree, we have reported that this species has really very high potential to mitigate the impact of climate change as well as the adapt the environmental conditions. Similarly, for medicinal plant species, so all these results have been published in SPR, Springer, or other journals also, which have already been submitted to the society. So, this will gives an idea that high elevated CO2 can be considered, in positive, because there is two ways. Either we have to leave it or either we have to stay in the condition. So what we can do, we can say use this high elevated CO2 concentration as a fertigation effect for increasing the raw material which is required for extracting the health promoting substance. So this is one example for what we have done. This is in case of Parthenium, how the Parthenium, different functional traits of Parthenium is being impacted. So if we expose the elevated this particular species to the elevated CO2 concentration, what we reported, leaf area was increased. It was increasing around 50% more than the control, and CO2 assimilation is 70% on part region. It is actually very, very, very high. In biomass production was also. And as I told you that nutrient limitation is a major factor which limit the plant growth and development under the elevated CO2 concentration. So we found that 300 kilogram nitrogen is required for proper maintaining the growth and development or enhancing productivity under the elevated CO2 concentration. Likewise, this is in case of poplar, where we have found that the Pseudomonas and Trichoderma were having very good result by enhancing the biomass production in this particular species. So these are actually few different mechanisms through which we can use as a biological measure to increase the carbon capture capacity of the terrestrial species, so that maximum carbon capturing is possible. And another one, which I told you that we are focusing on the urban species, how actually they respond. So what we did, same study we did in the urban area in different environmental gradient, actually, as I told you earlier. So we also reported physiological, biochemical, and biophysical adaptation. And what we reported, the most of the time, the vehicular emission significantly at the functional traits. So the photosynthesis decline, or the ROS formation was also there. Even we have also reported that under the stress condition, water efficiency was also increased. And how actually morphologically the plant adapt. One very interesting thing which we reported, under high elevated air pollutant, the plant increase the leaf thickness. This is just for the protection from the high temperature and the other air pollutants. So ultimately, we reported that these are the physiological modulation which determine the adaptation and resistance of this particular species. So this approach, actually plant function trait approach, can be used to screen the most effective or most better species, or for developing the urban green space, or developing the corridor, or developing the urban forest under the Nagar 1 scheme. So based on this research, actually we are also the member of different cities in smart city planning projects. Like Delhi, we are also the member in Dehradun and Lucknow also actually. So what we are doing, and based on this, we are also suggesting the species to different industries like Coal India Limited, Institute of India Limited, where they have high atmospheric, high air pollutant in their premises of the industrial area. So that we have screened and suggested the species, suitable species, so that air pollution impact can be minimized. And you can see from here, this is the picture from in Lucknow, where we are monitoring how actually the air pollutants alter the ecosystem process.
सर
This
आपने
is,
मारा
the
हमारा कोई और था
carbon sequestration.
नाम तो नहीं बताएंगे सर, बस आपको बता दिया आपने, गलत मारा हमको। संदीप बड़ा इंप्रेस हुआ, मार खाने के बाद मेरा नाम बताएंगे।
Sir, Dwivedi sir.
Yes.
Dwivedi sir, I think your mic is on, sir.
Yeah. So please switch on. Yeah. So how actually the air pollution or particular matter deposited on the urban canopy of the trees, they alter the ecosystem flow of ecosystem services. So you can see, with or without dust, how much carbon is being sequestered. So what we reported, due to the dust deposition or particular matter deposition on the leaf, we found that 40 to 60% reduction in the ecosystem services like the transpiration, cooling, or carbon capturing. And similarly for cooling, as I told you, this is also major question. Generally, people ask which species can create more cooling and which species can generate more oxygen. Actually, these are the major questions. And continuously we are being asked from the NGT or parliament, from the parliamentary question also, regarding the dust retention capacity and cooling and carbon capturing of capacity of the species. So this will give you an insight for the urban planner or manager who involved in policy making. So what we recommend that plant functional trait based approach has to be considered in urban planning policy, so that suitable or right species for right place has to be selected. Suppose that a particular species very well functioning in the commercial area or residential area, but same species may not be functioning well in the area where high vehicular interventions are there. So this approach will give you an idea to species to select a right species for right place, site specific selection actually. So this is also for other various species. Actually we have a screen for physiological biochemical responses and cooling capacity and dust capacity. And today's scenario, this is actually very important and major issue or pollution in the urban area. Artificial light at night. How actually this light can hamper the functioning of plant functioning and productivity. So this most of the time you might have seen actually some plants in urban area, they are directly exposed to light actually. So they disturb the physiological functioning. Even night also, this light can increase the transpiration rate also. So what we reported, the plant exposed to the direct light in night releasing more water. So enhanced water in such scenario may minimize the soil water availability. Then the situation will become water limited conditions. So already the water limited conditions are in urban area. But if you do this practice, then it will be the additional pressure to create the drought conditions. Okay, so one project is going on in this project. We are also, optimizing distance from the wall and distance from the light pole. How much distance should be from light so that there will be no impact on the physiological functioning of the plant. And apart from that, we have also, doing the study on the phenological trends, how the phenology is driving. So what we have report, significant achievement which we have made in investigating climate driven impact of tree physiology. And this alteration in the tree phenological will influence the ecosystem services, as I told you. So in India, we conducted this experiment in Dehradun urban area. What we reported that leafbud maturation advanced by 13 to 18 days actually. And similarly for leafbud emergence, subsequently leaf release occurred before 10 to 16 days. And after that, we did the global analysis through the meta-analysis actually. So globally, what we reported, temperature predominantly was found to be altering the phenological in temperate and boreal forest ecosystem. This accelerating the spring leaf out and flowering by 2.5 to 5.1 degrees centigrade. So this was published in Royal Society of Chemistry. One journal is in Environmental Advance. So this is the experiment which we did in FRI actually, just to see how the elevated temperature influence the phenological functioning or different phenotype of the plant. So you can see, so 13 to 18 days before early flowering we reported. So other, the significant achievement which we have made, but it is not covered in this, the presentation. We have elucidated carbon water dynamics in vegetation system through integrated ecophysiological and micrometeorological approach, just to understand the soil content, atmospheric content. So how ecosystem function and at what time ecosystem act as the carbon source or carbon sinks. This is actually very important to understand the carbon dynamics. And also we assess the vegetation net primary productivity through global dynamic vegetation modeling. LPG model is there through which you can assess the future climate change impact on the productivity. So that will decide whether your species or forest ecosystem will become, source or sink under the future climate change scenario. So accordingly, the manager or policy maker, they can develop some, policy or action plan to mitigate the impact of climate change. And apart from that, we have also, developed the greenhouse gas inventory from the different forest ecosystem, including agroforestry, wetland, grassland. On the particular aspect, we have completed 3 projects funded by the DST and 1 from the ISRO. This is the professional metrics based on the study which we have done. So over the experience of 17 years in the ICFRE system, our group has published, more than 220 publications, including 8 books, and we have attained around 6,600 citations with the H index 24. And as a PI and co-PI, I have completed 29 projects which were funded from the ISRO, DBT, DST, ANRF, and MOEF, NMHS, State Forest Department, consultancy service, and etc. So out of the 14 projects which I have completed in the capacity of PI, so this worth around, all projects worth around 39 crore which we have done. And I have guided 11 students, out of 4 as guide and 7 co-guide, and 90 is PG student, including MSc, MTech, or and I am also mentored 5 student, postdoctoral fellow from the DST and DST Women Science, DST Vice Fellow, and 1 research scholar under the CV Raman International Fellowship for African Scientists. This is one of the prestigious fellowship of ANRF. And I am fellow member of British Ecological Society and Association for Plant Science Research, and I membership of more than 10 society. And I have got Young Scientist Award from the DST, DVT, ACF Lux, ECOST, and Government of Uttarakhand, ICR, and the State Forest Department. So this is my actually the professional matter. That's all. Thank you, sir.
Sir, thank you for your presentation. Now we would like to call upon Dr. Rekha Kumari Ma'am, Department of Zoology, Miranda House, University of Delhi. Ma'am, you can start with your presentation. Over to you. Ma'am, kindly unmute yourself.
Oh, I'm so sorry. Oh, I'm so sorry. Good afternoon, everyone. I'm so glad to be here among such visionaries around. And I just heard Hukum Singh sir. He is a great achiever. And thank you so much to the organizers for inviting me for this talk. I would like to start my PPT, but is it me who has to present, or it will be presented from the organization side? I can also present it myself.
Ma'am, if you have the presentation, you can present it by yourself also.
Okay, let me do that. Give me a minute.
Dr. Jyoti, can I ask one question actually, please? Actually, right now I have a meeting actually, so can I leave it now?
Hello?
Can I present? My presentation is there. I'll just talk for 10 minutes, not even 10 minutes.
Sir,
Uh,
sir, is it
But
possible
yes,
for sir
actually-
to leave the meeting?
Dr. Praveen Sharma was to present. Actually, Professor Rekha Kumari has got some faculty meeting going on. And
Yeah,
she
I also
just,
came in between.
just took
I will
5,
take
10
only
minutes
5 to 7 minutes, not more than that.
Yeah.
Sir,
I think
actually, Hukum
Dr.
Singh
Praveen
sir,
will not mind.
sir, I think Hukum Singh sir was asking for a permission to leave the meeting, I think.
Yes, I think his presentation is complete, and we are so grateful for you to be here and present your work. Thank you, Professor Hukum Singh.
Thank you, sir. Thank you, sir. Thank you, sir. Okay.
Thank you, sir.
So can I start with your permission, sir?
Yeah, sure.
So, as Jyoti has already introduced, I am Professor Rekha Kumari and working in the Department of Zoology. And my lab is working in the area of molecular biotechnology and bioinformatics. So, I start with my academic qualification journey. I have done most of my education from my graduation till PhD in the University of Delhi. And I am also a double master's. I have done my second MS in bioinformatics from the University of Edinburgh with first division. And then my thesis was focused on the formation of rifamycin biosynth analogs, which was to combat the antibiotic resistance in the Mycobacterium tuberculosis. So my teaching experience is entirely in Miranda House. So I joined Miranda House in...
Ma'am, sorry to interrupt. Have you shared the slides? Because I think it's not visible on my behalf. I don't know whether everyone else is... Yes, ma'am. Now it's visible.
It is there?
Yes, ma'am. Yes, ma'am. Now it's visible.
Okay. So this is my this is the last slide I was showing about my qualification. So I can go ahead. So my teaching experience as well as my research experience is entirely at Miranda House. So I was just noticing that my previous speaker, they were talking about their research institute, but ours is mostly a teaching institute. We are teaching undergraduate students, but I am right from the beginning is interested in doing research. So I carry my research work side by side, and nonetheless, we have, done a lot of undergraduate research, which we have published also. I'll come to that later. But my total experience is right now at 23 years and 11 months. I'm going to complete my 24 years at Miranda House as a professor right now. So I have taught various papers at the graduation and post-graduation level, as well as other add-on courses. And I have been involved at in many activities at the university level, like most importantly, the syllabus revisions. So whichever syllabus revision have taken place in the last 24 years, I was always a part of it. I was also working between the DBT sponsored Science Setu and the undergraduate students and college students specifically through the program Science Setu, where we were motivating the students to come into the basic sciences. I'm also part of Indo-Dutch exchange program, which is run jointly by Miranda House and SRCC with the Dutch universities. I have been part of many conferences and symposia as an organizing member or even as a convener. In the college, I have till now, I have done most of the administrative work I have done is as convener. Right now, I'm convener of MH Vatavaran, which is the Environmental Society of Miranda House. I am also, taking care of the teaching of environmental studies, which is a mandatory AECC paper from the government side. And I'm also a founder member of a center. It's called the Center for Environmental Sustainability and Disaster Management at the Miranda House since 2021. I'm member of the Research and Development Cell, coordinator for the DBT Star College scheme. I have been part of NSS as an NSS program officer, worked at the hostel committee also. So I would like to talk briefly about the MH Vatavaran, which I am handling for last almost 5 years now. And this is a society which is taking care of many college initiatives or green initiatives, you can say, like we have our own paper recycling plant. If some of you remember that last time we organized a workshop in the college for SPR, we also provided our own handmade from the paper recycling plant, these writing pads and folders, etc. We mostly manage our waste waste. So we are towards a zero waste generating institutions. We have a composting unit. We have water conservation and rainwater harvesting reservoirs. We have energy conservation, and more than 70% of us, this electricity is also coming from the solar plants. We have an MH souvenir shop also, where we do sell these paper recycling plant products like pens or folders or bags, carry bags, and we have many other such products. Then we also have a plastic bottle revivals garden, as well as we have a lot of other collaborations like with Bisleri, where we give all our plastic waste and they give us in return, very utility items like benches, etc. for the students. We have won a lot of laurels for MH Vatavaran. For the first time, this centenary cup was established by the University of Delhi, which we won for MH Vatavaran, as you can see it here. And the activities like plantation drives, cleanliness, we are always celebrating this World Environment Day. And there are many other activities like awareness workshop, etc., which we carry on. MH Vatavaran also takes out its annual magazine called Madhuban, which contain mostly the articles given by the students and teachers alike. So again, these are some of the accolades for the MH Vatavaran. We have been certified by various national and international agencies as the top institution in the waste management. So as I was a program officer in NSS, again, I was involved in many outreach activities like teaching of the slum, the underprivileged kids, Sadbhavna, where we were moving to, we were contributing and going to volunteering to the old age homes, even the orphanages, etc. I'm not going into details. Then there are a lot of co-curricular activities which I am involved for last so many years. So we have this Inspire internship program. We do a lot of capacity building workshops. Then DBT summer research workshop, which we are going to start even this year from 9th of June. Then we have a lot of bridge courses, research lab visits, educational excursions, etc. Apart from this, I run two ad-hoc courses or short-term courses, which are certified courses. They are self-financed courses also, where we are training students for the skills used for the medical biotechnology, as well as the bioinformatics and biostatistics skill sets. Then these are some of the, again, like incentives. I also was awarded as the Teaching Excellent Award for my research innovation projects, which were sponsored by the Delhi University We worked on, again, antimicrobial compounds and looking at the resistomes in the soil. Then I have done, and I have also organized of refreshers, conferences, workshops, etc., given invited talks. And these are my international achievements. Like, I have been awarded as Vice Fellow from 2009 to 10, where I worked at the Department of Fundamental Microbiology, Lausanne, Switzerland. At this time, we made a microbial biosensor for the detection of the oil. This project essentially was, focused to detect the presence of oil in the sea. When there are like oil spills, you know that the oil tankers are crossing, they are coming through the sea route. So sometimes there are oil spills, unavoidable, and if you and this is going to harm the marine life. And to detect these oil spills at the source, we made a microbial biosensor which can detect the oil even at the nanoliter or nanogram. It was highly sensitive, which we published also. Then I was on the faculty training program to University of Edinburgh, where I did my MS in bioinformatics. Then also I worked on the embryonic stem cell regulation. Then I was also awarded through INSA DFG a fellowship for as a visiting scientist at the university, this Center for Environmental Studies. It was ZF Zurich. It was in Leipzig, University of Leipzig. And these are this is the very short list of like what all projects I have completed. So I have done work mostly in the environmental pollution area, green nanoparticles, etc. And then right now we are working on these environmental pollution like bioresource and health. I have 3 PhD students at the moment which are enrolled with me, and 1 postdoctoral. And one of the students is working on the bioresource and the human health. She is like not just we are like looking at the quantity of this, but we are qualitatively also doing the analysis of these bioresources using the this metagenomics approach. Then we are working on landfill soil pollutions, which is again a hub for potential biodegradables. And another project which we are working on is the impact of water pollution on human health, especially near the landfill sites. And this is a DBT sponsored project in green nanobiotechnology and human health, where a biocare fellow is working with you. So talking about my first ongoing project right now, it's a PhD project also. So it's air pollution, bioresource, and human health. We all know that the air pollution is not a it's not a joke anymore. It's a very very important thing. It can always choke us. And these bioresources are of very high concern to us, because these are not only the like dust particles. They also have the biological entities like viruses, bacteria, even endotoxins, etc., etc. And they can be responsible for various diseases, and especially the respiratory diseases, as we know that a lot of people are suffering from asthma. They are lung diseases, etc. So we have this cascading factor which we use to even segregate these bioresources by according to their size, and look at how specifically these size is important, and they can impact which part of the respiratory respiratory system. So again, this is the metagenomic approach and the modeling of these bioresource deposition in the respiratory tract. Another, as I said, we are also working on the water pollution. And this water pollution, as you know, especially at the landfill sites, is because of the leachate, which is, it's very, very dangerous, especially during the rainy seasons. It leaches out, and along with it, it brings a lot of pollutants. And this, we did a study on the water quality assessment in the academic institution. We just got published in the scientific reports, and here we were talking about the how important it is to keep doing the monitoring of these water sources which are in the academic institution, because we are all mostly our time is going at our institutes, and if they go unchecked, it can lead to many waterborne diseases, and the, when like occupants will not be at ease. Then the landfill leachate and health risks, as I just said, that these leachate is a very, very important source of lot of parasite pathogens which we have to take care of, and we have all kind of approaches we use, like physicochemical analysis, physicochemical pollutants we look at, and then we also look at the chemical pollutants as well as the biological assessments. And right now we are working, and this also includes survey-based assessment of the health conditions of the residents who are living near these landfill sites, so that we can complete the story, what we are looking, what we are monitoring, the kind of pollution level in the source, like the the water there, and how it is impacting their health can be completed. This is another project. Again, we are looking from the landfill sites. The soil samples are screened for the presence of these biodegrading, potential biodegraders, so that we can use them for the remediation of pollutants like pesticides. Now, this is the project, as I said, which is looking at the antimicrobial drug delivery systems against AMR and cancer, where we are making the green nanoparticles using Triphala, Giloy, and neem. And we are looking at their potential as antimicrobial agents and how effectively they can be delivered to the patients. So this is methodology. I'm not going into details about it again, and maybe if somebody is interested, we can talk about it later. And this is the my recent publication in the last 5 years, and I have a cumulative impact factor of 20.7, at almost 55.4 since the beginning. And we have, as I said, recently published in scientific reports. Before that, I also worked on these biochemical profiles of the pollen allergens from SPR, where we did a pilot study to know that how allergens and their chemical properties can lead to allergies, and allergens can impact the immune system of the humans. These are some of the quick chapters I have written so far, and this is my lab. We have a lot of other activities also, like I work basically at graduation, undergrad, post-graduation, and all levels, not just the PhDs or postdocs. So because I'm in an institute where the teaching is the mandate, but now it is the research which is again, taking up. And since I'm interested in research from the beginning, so I'm continuing with my own lab. Thank you so much. If you have any questions, I'll be happy to answer.
Thank you so much, ma'am, for your presentation. As I think we are running out of time, so I would like to invite Dr. Praveen Sharma, sir.
Yes, sir. Yes, madam.
Sir, kindly, you can start with your presentation, sir.
Okay. Cancer?
Entire screen there, sir.
Huh?
Entire screen.
Thank you very much, Professor Rekha, for wonderful presentation. Since we are running short of time, I would request Professor Praveen Sharma to kindly start his presentation. Thank you.
Slides.
Okay, my slides are visible now.
Yeah,
Yes.
they are.
Yes, sir.
Okay, myself, Praveen Sharma from Himachal Pradesh Agriculture University. Regarding my introduction, Praveen Sharma from Chaudhary Sarvan Kumar, Himachal Pradesh Krishi Vishwavidyalaya, Palampur. I did my PhD in vegetable science with the minor subjects plant breeding and genetics and plant pathology. I cleared this NET qualified in horticulture as well as in vegetable sciences. Presently, I am working as a professor, and I joined here in the university as assistant professor. Before joining assistant professor, I also served in private company, that is Indo-American Hybrid Seed Company. My areas of specialization are vegetable breeding and genetics, then development of varieties in vegetable crops for the protected cultivation against the diseases, soil-borne diseases like bacterial wilt, and development of the different varieties in cucumber also, that is parthenocarpic type of cucumbers. You can say the seedless type of cucumbers. Then I also developed technologies in soilless cultivation of vegetable crops like hydroponics, and also did research on vegetable grafting, because in Himachal Pradesh, soil-borne diseases are hampering the yield of the vegetable crops. So grafting is very important. Regarding my research contributions, I developed one hybrid of tomato that is resistant to bacterial wilt, and for your knowledge, this hybrid is resistant to bacterial wilt and was first in the package of practice of the Himachal Pradesh. Apart from this, I also developed two varieties in parthenocarpic type of cucumber, that is Him Palam Khira 1, Him Palam Khira 2. And in addition to this, I also developed two cherry tomato varieties for the protected cultivation, that is Him Palam Cherry Yellow, Him Palam Red Cherry. Then in addition to this, I also developed one variety that is of capsicum, that is Him Palam Yellow Capsicum. Apart from this, as a main breeder, I also contributed as a co-breeder in the development of different type of varieties in Himachal Pradesh, like your Himachal Lobia, then in bees, in turmeric, in chickpea, in tomato, in chili. Then in addition to this, I standardized 8 production technologies on the protected cultivation, which are now included in package of practices of the university. I published more than 200 research papers in different reputed journals of NAS rating also. Regarding the research projects and funding, as principal investigator successfully completed 30 ad hoc projects as principal investigator, funded by Indian Council of Agriculture Research, RKVY, Department of Science and Technology, Bird Wing, etc. The key projects included protected agriculture and natural farming, that is Bird Wing funded project was there, in which in that project I was procurement officer as well as component PI. Then development of the disease resistant hybrids, hydroponics, and soilless cultivation of the vegetable crops. Regarding the students' academics, regarding student guided, I guided 10 PhD student, 21 MSc student, and I am in the advisory committee of 30 PhD students, 37 MSc students. And I also developed new courses and practical manuals on vegetable cultivation as well as protected cultivation, and conducted trainings on GRF, NET, ARS, etc. Regarding the administrative and leadership role, I was procurement officer in handling 18 crore Bird Bank ad hoc project. I also acted as GDO of the department, principal investigator of number of projects, then member of All India Coordinator Research Project and state level committee, then organizer of the national training programs and workshops related to the protected agriculture. I am also president of the Teacher Association, and I was president of the Agriculture Scientist Forum, and presently also handling the charge of the farm in which students and scientists are doing research. Regarding the awards and recognition, I elected as fellow of the Indian Society of Vegetable Science in 2023, and selected and completed advanced training, and that is 3 months training on vegetable production and post-harvest management at Israel, which give me the exposure to do the research under the protected environment and hydroponics also. So among the research, you can see that photograph of the attractive fruits which I developed, that this hybrid is completely resistant to the bacterial wilt. Otherwise, chemicals are not working against the bacterial wilt incidence. Only the resistant gene is working in Himachal Pradesh and other areas also. You can see the shape and size of the tomatoes of this variety, and this variety is very much popular among the farmers of the state. You can see the training of the different agriculture officers under the protected environment, under the polyhouses, for the cultivation of tomato in our university, and the technology which we are transferring to the farmers is validated under the protected environment. You can see this is the new variety which is indeterminate in habit. You can see the first photograph that this variety is taller than human being also, and this is him palm yellow capsicum. Again, this is a solanaceous crop, and this is also susceptible to the bacterial wilt, but this variety is resistant to the bacterial wilt and gave yellow color fruits. This again is very much popular among the farmers. This is the long procedure to how to develop the varieties. It takes, I want to show that it takes more than 11 generations to produce a single variety. So this is a very laborious work which we did in the university. And you can see the green fruits of the capsicum variety. The characteristics of this fruit is that this is 4-lobed fruit, and this is very much popular among the farmers also. And the durability, or you can say the shelf life of the fruits, is very good. And this is the technology which we developed for the production of this. And this technology included the seed rate, the spacing, the initial dose of the fertilizer, then the fertigation, etc. And the variety is, I again telling that this is resistant to the bacterial wilt. Similarly, we developed this cherry tomato varieties of the both color, that is in red color as well as in yellow color. And this is again the procedure took more than 11 years to develop a single variety of cherry tomato also, because cherry tomato is again very much popular among the 5-star hotels also. This is the photograph of cucumber variety. That this is Him Palam Khira 2. This is a seedless variety of cucumber, and very sweet and tender and very much popular among the farmers field. And you can see again 11 years hard work is there to develop this variety. And apart from this, a dark green color cucumber variety was also developed by me here in the university for the farmers of the Himachal Pradesh. Now these all varieties are included in the package of practices for the farmers in the university. Now you can see the development of tomato. We also developed tomato through grafting under protected environment. You can see this is a single plant, and in the roots you can harvest potato. On the same plant, in upper portion, you can harvest tomato. So this is the wonder plant, and very good popularity among the farmers field. Again, this is our success story also. Apart from this, I also gave some recommendations on seed rate of these high value vegetable crops, that is tomato, capsicum, cucumber, etc. And their spacing, their cutting system, their pruning system, etc. You can see the recommendation of lettuce in the hydroponic system. We tested number of varieties, but concluded that in hydroponic system, in hilly areas, in our areas, the leafy type of this lettuce is very much popular among the farmers. And we also imparted the training to the agriculture officers as well as to the farmers for the cultivation of this. My students are also working to develop the varieties of parthenocarpic cucumber and lettuce for the hydroponic system, soilless cultivation, because there is a lot of pressure to the soil on the soil, and number of soil-borne diseases and pests are there. Now our focus is to develop the varieties which do not need the soil. We can grow soilless these crops in soilless media. This is the popularity of our students, that they develop their own business to grow this hydroponics, to grow the vegetables in the hydroponic system and supply to the metropolitan cities. And our work was also recognized by the Governor Himachal Pradesh, Honorable Governor of Himachal Pradesh, you can see, along with the Vice Chancellor also. And I also took training in the Israel for more than 3 months to develop the varieties, as well as to procure the fruits under the room temperature for more life. Thank you very much. This was only the highlights of my work. Thank you. Dhanyawad.
Thank you so much, sir, for your valuable presentation.
Thank you.
Now, I would like to call upon Dr. Sunita Mahapatra, ma'am, from Mohanpur. Ma'am, are you available here? Dr. Sunita Mahapatra, ma'am, I think she's not here. So now we can call upon Dr. Anath Bandhu Das, sir, from Utkal University, Bhubaneswar.
Yes, absolutely. Yeah.
Sir, you can kindly proceed with your presentation.
Yeah. So just I'm sharing my slides.
Okay, sir.
Is it visible right now?
Yes, sir.
Yeah. Audible also?
Yes, sir, audible.
Yes.
So I think your slide is not.
Now it is visible.
Yes, sir.
Yeah. So good afternoon to everybody, and at the outset, I would like to thank Professor Bhatnagar for selecting me as the awardee of this prestigious award, Fellow of SPR. So, in fact, already it is late, but still then I will try to finish my lecture very quickly. In fact, I did my BSc and MSc and PhD in Calcutta University under the able guidance of Professor A.K. Sharma and Oksana Madam. Then after I moved to Odisha in Forest and Environment Department as a scientist. Then after I went to Utkal University, this OUAT, that Odisha Agriculture University. Then after moved to Utkal University and in Department of Botany, and still I am continuing there after the retirement as emeritus professor. So since this university is a very old university and first university of the Odisha, and Honorable President as well as our Education Minister, all are the alumni of our university. And since Hukum Singh sir actually proceed some of the things of my research, what I did for last few years because of this global climate change and frequent visit of super cyclones we started the experiment on the mangroves, salinity stress of the mangroves. And in that salinity stress, basically we worked on the physiological responses, biochemical responses, and molecular responses, different type of responses we did. And of course, all these mangrove populations will be different if you look into that, because as you know that the mangroves are inundated twice in a day, and they have a high salt pressure there itself, and you can see that they are escaping themselves from the salt with the help of salt gland, as well as new metaphors are there, because this is one of the plant Aegiceras corniculatum, where very prominent salt crystals are generally observed on the top of the leaf. And when we proceed some of the research, we could see that some of the other mangroves like Swedian odiflora, they are adapted themselves as per the salinity. And this, because my basic background was genetics and plant breeding, then after I switched over to molecular physiology, so that from the chromosomal perspective, I could see that Soidia is changing its chromosome number as well as the ploidy as far the salinity is concerned. So in the high saline area, they have adjusted their ploidy level as compared to the low salinity area. And similarly, we experimented some of these three mangroves, Brugueria parviflora as well as Aegiceras corniculatum, and we could see that there are variations in the antioxidative defense system, like catalase, peroxidase, as well as vehicle peroxidase, SOD also we have, and we published in very good journals. And in fact, the salt stress of the mangroves has given me a lot of citation. One paper has given about 6,000 citations. And also we have looked on the pigment protein complex in the thylakoid, and we could see that in the jet stream, all these thylakoid proteins like cytochrome B6, B12 complex, and all these other protein complex also varying as per the protein is concerned. And so that the thylakoid membranes actually get damaged in the latter stages. We could see that there are specific salt stress marker, which is 23 kDa protein band. And this protein band, generally we could see that these protein bands are localized in the the thylakoid membrane, as well as so that the membrane bound protein generally is 23 kDa protein bands are nothing but a membrane associated protein, which is actually hamper the thylakoids as well as the structure of the thylakoids in the high saline condition. So, and later on also some of the other scientists, they have isolated some of the salt stress gene and incorporated with Arabidopsis thaliana, as well as our Professor Ajay Kurida also did some of the work in MS Amina foundations where the salt stress gene has been incorporated in vocally variety of the rice. And then after we started some of the work in the climate resilience rice system, where Hukum Singh ji also worked the same line in the forest plant, we actually screened with the aim of the high CO2 concentration in the coming days, which is expected in 2050. And we screened 16 superior genotypes in open top chamber. And we could see that because in 25% carbon dioxide concentrations, which is about equivalent to 470 ppm or 660 ppm, the highest one, that 75%, that in the high concentration of the sorghum variety, we could see that there are the growth, high growth in the biomass, both above ground biomass and below ground biomass. They have the increased concentrations with the carbon dioxide, but unfortunately, this carbohydrate which is produced during this high CO2 for fixation of the CO2 that is not converted into the grain. So grains are chappy. So that's why it is a very concern that in high concentration, maybe most of the variety which is nowadays cultivated in Orissa may not produce that grain. Instead of good grain, they will be having the chappy grain, except few varieties there. The Rubisco is very highly developed and they can actually manage this high CO2 concentration as well as temperatures. Here you could see that the root biomass also increased, but out of these 16 variety lollards are much more suitable to adapt. Sorno is not so suitable, or Sorno Sarban. Khandogiri to some extent it is working very well as the grain number as well as the grain yield is concerned. And also in the seedling stages also we have started this experiment in the outer field as well as in the open top chamber as well as in the climate control greenhouse, and we could see that this Lollard variety is working very fine as compared to Khandogiri or some of the other newly developed variety. Besides this, also we have started and did some of the work in the silicon uptake mechanism, because most of the cases we could see that silicon has a greater role to play in rice for the protection purpose. And we could see that that silicon can prevent physiological stress, chemical stress, as well as enhance the biotic stress. And so that we started some of the work on that silicon uptake mechanism. And in the low silicon uptake variety, generally they are actually hampered with this insect population as well as some of the fungal populations. Also, they can attack very well. But uptake of this variety, you could see that silicon concentration about 2 ppm is maximum, which can millimolar is maximum, which can be obtained by different varieties. And there we could see that your silicon transporter gene, one is SIT1 or and SIT2. Two genes are transporter genes are there. One is responsible for the transport of the silicon from external to the epidermal cells. Then after from the epidermal cell to the xylem, transport is done by the SLS2 gene. And we could see also that the silicon deposition in the field also, that diatomaceous starch 2.5 kg per hectare can give much more silicon deposition as compared to this rice husk as 2000 kg per hectare. And as compared to the control, they are generally giving much more protections to these plants, but DAE is quite high in cost as compared to the rice husk as. And also we localize the LS1 gene, LSI1 gene, which is a transporter from outside to the inside with the help of this anti-LSI polyclonal antibody into the xylem. And also later on, some of the scientists, they could localize that LSI1 is found in your rice chromosome, and which is about in between this 16.9 to 1.6 your centimorgan distances. So that we started some of the experiment in screening the varieties, rice varieties, and we could see that there are differential uptake of the silicon into the rice, and also we could see that we have developed some primer out of this LSI2 gene, which is a transporter of epidermal cell to the xylem. So, and we could see that some of these very good amplifications of this LSI2 gene in some of these rice varieties, we could see like Annapurna, Badami, etc. And then after your sequence alignment we did, and then we could see that this SI transporter gene in contrast is one of the unique gene which is present here itself. And with the help of this Ramchandran plot, and we could confirm that yes, this contextually is one of the gene where the LSI2 gene is quite remarkable, and they are much more robust as compared to the other varieties that we have started. So that from that, because this LSI2 gene are generally transporting this silicon and giving them much more protection. And so beside these, also we started some of these work in the wild banana in Northeast area. And we could, because mostly we worked on the diploid banana where it is producing seeds. As you know that the banana of A-type, B-type genomes, generally they are mixed naturally in natural conditions. So all these seeded banana having the 22 chromosomes, they generally germinate to produce the seeded type of banana, which are much more useful for the breeding program of the banana. Then after we did a large number of the chromosomal work of this northeast banana and very tiny chromosomes, but it is very much useful because we learned the technique from Professor A.K. Sharma. Then after I have done this fluorescent in situ hybridization with both the pioneer worker from Hislop Harrison's laboratory as well as Professor Dolly's laboratory in Czech Republic and UK. And also I did some of the work with the help of the DBT fellowship with Kew Botanical Garden as well as Queen Mary College of London. And we could see that these genotypes are Indian genotypes, yellow color patches or these green color patches, they are, or these red color patches, they are the unique genotypes. When we compare these genotypes with the global banana genotypes, because I am the member of the global banana genotypes, which is maintained in the Leuven University in Belgium. And also we started some of the work in the local banana, about 10 varieties of the local banana bodies where the tissue culture work we did, and it has been established and it is very much successful story for giving all these tissue culture banana to different 30 districts of the Odisha by the OTLP program of government of Odisha. And then after we started some of these work where the Fusarium oxysporium is one of the major threat for the banana for the Panama wilt. And we started some co-culture of this Fusarium oxysporium and Trichoderma horgianum, and we could get some good results that Trichoderma horgianum can suppress this oxysporium to some extent in the low doses. So that we did some of the work and also physiological work. We did some of the things that were about 20 microliter of this what we call Fusarium, which is about 6,000 spore load, can give it can be tackled by this Trichoderma, whereas about 12,000 doses of the spore cannot be tackled by this Trichoderma. So these are some of the work we did, and also some of the physiological work where this photosynthetic efficiency, how it is checked with the help of this one. Because in the K bands, for example, there, if you are looking to that oxygen evolving complex is choked with the help of this Fusarium, whereas Trichoderma can normalize that one so that the photosynthesis can be worked very well. And also later on, we started with the help of this training of Professor Hislop Harrison, so where the transposable element we did some of the work in banana, and we could see that there are some transposable element which is very much found in some of these A type of banana or B type of banana. A type of banana is basically tabletop banana or ripened banana, whereas B type of banana are cooking type of banana or plantain banana. And we developed, because we could see that some of the transposable elements, they are moving from one place to another, which generally gives different varieties of the banana. And also we could establish with the help of this fluorescent in situ hybridations that A-type banana and P-type banana, with the help of that one, say for example, Pisang Raja, where the A-type, double A and B-type is there, whereas triple A type, where all these red spots are nothing but a hat one, is one of the transposable elements which can be located like that. So more or less, this is in nutshell the work. And in the meantime, we did. I produced about 19 to 20 PhD, 2 BSc, and about 65 of MSc student, and also with the help of the DBT as well as some other funding agencies. I visited abroad, Netherlands as well as your UK as well as Scotland, then Japan also, and I received some of the awards like some of the Chandrasekhar Award from the Odisha Vigyan Academy. Then after Hiralal Chakraborty Award from this Indian Science Congress, and of course this fellow of SRP is one of the your prestigious awards. So thank you very much for giving the opportunity, and also thank you very much for your Jyoti ji as well as Professor Bhatnagar. And also recently I am in the editorial board of the Vegetos, as well as I am the chief editor of Odisha, your Botanical Society, one journal is there, about 35 or 38 years old journal. So, Journal of Plant Research. Thank you very much for giving this opportunity.
Thank you very much.
Okay.
Thank you very much, Professor Das, for your wonderful presentation. And I am sure that Society for Plant Research and Vegetos will be benefited by your vast experience and a very dedicated methodical approach of working. Thank you very much for being with us.
Thank you very much, sir. I will be.
Thank you.
Thank you, sir.
Sir, may I leave right now, sir?
Yeah, we are having just a brief address by our president,
by
the national president, and then we will conclude.
Okay, sir. Yeah. Okay, sir.
So thank you, sir, for your presentation. Now, may I have the distinct honor of inviting Professor Dr. Banna Bose, ma'am, our national president of SPR, to deliver the special remark and kindly bless the SPR fellow awardees with her inspiring words. We are truly privileged by your gracious presence, and I humbly request you to address and bless the awardees, ma'am. Ma'am, over to you.
Okay, thank you. Am I audible, sir? Am I audible? Yes.
Yes, ma'am.
Good afternoon to every distinguished persons present in this webinar. I pay my regards to Professor R.R. Hanchinal, Chief Patron of SPR, and Professor S.K. Bhatnagar, the founder chairman of the society, for convening this most important webinar regarding the presentation of fellow awardees of SPR for this year. I also grateful to Dr. Dolly Wattle, the chairman screening committee, and every member of the screening committee for their valuable work, for which these commendable results come in proper platform. However, we heard the presentation of fellow awardees one by one. Those are very interesting and which educate us about their valuable works as well as about present era of scientific highlights. I congratulate all the awardees for their great achievement from SPR. Finally, I pay my gratitude to all EC members for their presence in the webinar of SPR. Lastly, I am very much thankful to Jyoti Singh, moderator, and Dr. G. Narayan for their valuable work for for holding this webinar. Lastly, I may add that today I also passed a very, very valuable time with all of you. Thank you very much and wish a great success to SPR and the journal visitors. Thank you to all of you.
Thank you very much. Thank you very much,
Thank
madam, for your kind words. Yes, Jyoti, kindly
Thank
extend
you
vote
very
of
much,
thanks.
ma'am.
And
Yes,
then-
sir. Yes, sir. Ma'am, thank you for your blessings and motivational words for the awardees. Your valuable guidance, encouragement, and visionary thoughts have truly inspired all the young researchers and the scholars present here today. Now, on behalf of the organizing committee of the SPR Fellow and Best Doctoral Thesis Award Lecture Series, I extend my heartfelt gratitude to all the distinguished dignitaries, eminent scientists, academicians, researchers, scholars, and the participants who joined us today from different parts of India and abroad. We express our sincere appreciation to all the presenters and awardees for their insightful and inspiring lectures, which greatly enriched today's session through their valuable scientific contributions and experiences. I would also like to convey my special thanks to our respected chairpersons, moderators, members of the organizing and screening committees, and the technical support team for their constant and smooth coordination in making this event successful. With this, we conclude today's session and look forward to your continued support and participation in the upcoming session of the lecture series. Thank you once again, and wishing everyone a pleasant evening ahead. Thank you.
Thank you all. Thank you very much.
Thank you. Thank you very much.
Thank you, sir. Thank you.